Thank you very much. Good evening in China. I'm going to present a quick account of Artemia genetic resources in the world. Next, please. What I would like to highlight here is that we have 6 regional sexual species and also a lot of asexual types distributed in the world. But what is most important is that most of these genetic resources are harbored in locally adapted populations. And Artemia franciscana is a very well example of that. I will talk about that later. And also we have incipient farm types, as it was mentioned before. Next, please. Well, what are the challenges? One is lack of knowledge to assess climatic change and other perturbations. There is another problem that the genetic variability is losing at alignment rates. And you know that genetic variability is really important for species or population resilience. So this is a worrying trend. There is a need of systematic monitoring of genetic diversity. This is to comply with international guidelines, FAO, for example, and CBD, that means Convention of Biological Diversity. And I think, and this is to be discussed, there is no policy or institutional framework for monitoring and protecting wild intraspecific genetic variability. Next. Okay, these are the 6 species. You know them very well. Artemia persimilis is the oldest, and we are lucky to have it in Chile and Argentina. What is important here is that each species is a unique combination of genes and interaction with the environment. Each has a unique evolutionary story. So I think that one of the main problems is that franciscana is the best studied one, but we need to study all the other species very well because we are to find novelties in each one. On the right side, you see the tree that only shows that using different nuclear or mitochondrial markers, okay, you get always the 6 species. There are some populations that normally do not match with the species, but this is something that we could discuss in another opportunity. So what is important is that all the populations are locally adapted. This is the example of Artemia franciscana, the most widely distributed species. You see here on the right, there is a wide latitudinal variation. All populations are different genetically and phenotypically. And that means that maybe they have aquaculture traits or adaptive traits that are unique, but we need to discover them. The other point that was already mentioned is translocation to different places in Macau, for example, Brazil, Vietnam, China, Thailand, I saw Kenya. And this is very important because we have the origin of farm types. This is something that we need to continue monitoring. What is the difference between a farm type and a local wild population? And what is really important to understand what means locally adapted population are the studies on genomics and transcriptomic studies that we hope will provide clues on adaptive and productive genes. Okay, this is the Vinh Chao farm type, Vietnam, that came across after San Francisco Bay cysts were introduced in Vietnam. In 1996, I saw from Professor Sorghilu's presentation that cysts were introduced in Macau, in Brazil, earlier on. So we don't have much information about the quality of those cysts. What is important here is that selective pressures in Vietnam selected individuals with tolerance to high temperature. And what is known so far is that heat shock proteins are upregulated, are differentially expressed. But as you will see later on, there are so many genes that can be upregulated depending on the environmental conditions. Next, please. Yes, thank you. So here you have the environmental component of the reproductive mode. I consider this because of the importance for survival of the species and populations, but also because it has a lot of productive importance. On the genetics side, well, you will see later from Gilbert, Artemia genome has a lot of genes, as many as humans. So I was impressed to see that. So you have a very complex, you see in the bottom of the figure, genetic environmental interactions. And this is really a black box that we need to understand. And I think Artemia is a very good material to do this, even under laboratory conditions. On the left side, well, the genetics of the mother could be important in perceiving the environmental change. We, in very early stages of the genetic knowledge of Artemia in 1998, showed that females with multiple genes with high variability or heterozygosity were able to produce more cysts. So you see here the genetic makeup influences the ability to produce cysts. So I see with good eyes the managing work that Thomas is doing in Great Salt Lake to maintain the ability of females to produce cysts. But what you see in the third line on the left, there are more than 300 genes controlling cyst diapose and post-diapose functions. That's a lot. If you see the interaction of each gene with a particular environmental condition, you will be amazed the difference that can appear. For high salinity tolerance, there are more or less 600 genes, and this is also impressive. And for anoxia tolerance, about 900 genes. So if you think in the interaction of these genes with the environment, okay, we have a great difficulty. So this slide is mainly to convince you that we need to improve the characterization of different wild populations of Artemia and also of farm types. For example, what is the difference between the Vietnam strain and the Macau strain? Next, please, Rodrigo. Okay, what are the problems? Well, we have a mandate to protect Artemia natural habitat and their biodiversity, because these resources are source of novelties for aquaculture. But what is happening the saline lakes are declining around the world. As you see here, they are shrinking at alarming rates. And Thomas mentioned about Great Salt Lake. The photo on the left is Great Salt Lake, published by this author. And then you have Urmia Lake. I think, Patrick, that we did, we took that boat when we were in Urmia. So you see the boat there. And also it's very interesting to see what is happening in Ariel Sea. Thomas talked about that. So next, but as important as the loss of species is the loss of genetic variability. So I put here a graph that I think is very important because it illustrates us the type of information that we need to have. Here you have a contemporary heterozygosity on the left, and in the bottom you have historical heterozygosity. That means that they collected the ancient DNA. And over the line, you have the genetic diversity gains. And below the line, you have genetic diversity losses. So what is important, if you see all these species that are here on the top with different kind of classification according to IUCN, the conclusion is that they are losing genetic variability. And if they are doing so, they are losing their ability to persist in the environment. Next, please. One of the studies I know, besides Great Salt Lake, that is being monitored for more than two decades, is Artemia urmiana in Urmia Lake. So after 10 years, next please. Oh, sorry, it didn't appear, but author showed here that polymorphic loci are losing variability after 80% of reduction of the surface of the lake. So finally, next please. I think it's important what I said regarding establishing, strengthening national global characterization, monitor, and information system for Artemia genetic resources. This is a mandate of FAO for all species, and we really hope that you realize that we have very few information about all the populations composing the different Artemia species. It's important to have information for detecting diversity losses over time. And here is very important to have cyst collections from around the world. So it's very important what Li-Yin Su is doing with the Artemia reference in Asia, and also what the Artemia Reference Center in Belgium has done in the past. We need to assess differences between farm types and local populations, and we need to coordinate actions to comply with international guidelines. I think the Artemia genome that is recently published will facilitate the study of intraspecific genetic variation, identification of key adaptive genes. I mean, there is a very good opportunity to do genotype environment interactions, and Artemia is a very good model to do that sort of studies in the laboratory, and also could be done in the wild. Okay, thank you.
you.